Having covered genes that promote and control cell growth and regulation, we're now going to move into those genes whose predominant role is to suppress the development of tumors. Tumor suppressor genes are genes that encode proteins that regulate and suppress cell proliferation, predominantly by inhibiting cell progression into the cell cycle. Some of the molecules that are involved in this are things such as p53, which prevents a cell that has damaged DNA from entering S phase and induces the creation of DNA repair genes. And we've discussed one of these already, the retinoblastoma gene, which prevents cells normally from entering S phase until appropriate growth signals are present. The inactivation of tumor suppressor genes requires us to discuss for a moment Nunsen's two-hit hypothesis. Nunsen hypothesized that if there are two copies of a tumor suppressor gene present within the cell, both copies must be knocked out in order for the cell to develop into a cancer. The first hit can be inherited germline or be an inherited somatic mutation. The second hit is generally an acquired somatic mutation. Examples of inherited germline mutations can be seen in the retinoblastoma gene, where patients have familial forms of retinoblastoma due to germline mutations of the retinoblastoma gene on chromosome 13. Also the Li-Fraumeni syndrome, which is due to germline mutations of the p53 gene on chromosome 17. The cartoon in this slide simply demonstrates the difference between the familial and sporadic form of retinoblastoma, both of which demonstrate the need for Hudson's two-hit hypothesis. Here, in both cases, both forms of the retinoblastoma gene are knocked out. However, in the familial form, the child inherits already a mutated form of the retinoblastoma gene, which has been knocked out in one of the parent's gametes. The child then undergoes a second somatic hit of their other form of the retinoblastoma gene, and with both copies of the tumor suppressor gene knocked out now, develops retinoblastoma generally bilaterally and early in life. In the sporadic form, the child inherits two working copies of the retinoblastoma tumor suppressor gene, one from the father and one from the mother. As they grow up, the child must undergo two somatic hits, one that knocks out the father's version of the tumor suppressor gene and the other that knocks out the mother. This child will be more likely to develop a unilateral retinoblastoma and one that develops later in life. Remember that with retinoblastoma locked out, there is E2F transcription and progression of the cell through the cell cycle. Another major player in the realm of the tumor suppressor genes is p53. It is mutated in over 50% of tumors, including those of the lung, colon, and breast, and its predominant role is in response to DNA damage, whereupon p53 is phosphorylated, causing the cell to arrest in G1 and leading to the induction of DNA repair genes. If the DNA damage cannot be repaired, p53 directs the cell to undergo apoptosis. This is another tumor suppressor gene, the APC gene. It is found on the long arm of chromosome 5, and in families where APC is mutated, there is the development of what we term familial adenomatous polyposis, which will be discussed more in the GI lecture. APC usually involves in breaking down beta-catenin. As you see here in this cartoon, when APC is not present, beta-catenin can translocate to the nucleus and upregulate transcription. Other tumor suppressor genes that one may need to know about include the von Hippel-Lindau gene, which, when both forms are mutated, leads to the development of von Hippel-Lindau disease, which demonstrates the development of tumors such as renal cell carcinoma and hemangioblastomas. The Wilms tumor genes are also tumor suppressor genes, as are the genes involved in the development of neurofibromatosis type 1 and 2. Neurofibromatosis type 1 characterized by the development of numerous neurofibromas, neurofibromatosis type 2 by the development of acoustic neuromas and meningiomas. We've also mentioned some of these other tumor suppressor genes in our other lectures, such as in the lecture on breast cancer, where BRCA1 and BRCA2 are tumor suppressor genes that, when mutated, lead to the development of breast and ovarian carcinomas. Having dealt with mutations in genes that lead to uncontrolled cell growth, both the oncogenes and inactivation of the tumor suppressor genes, we're now going to move on to looking at alterations in genes that regulate apoptosis. Apoptosis is a process which is carefully regulated both by pro- and anti-apoptotic factors. Some of the factors that prevent apoptosis include BCL2, which is overexpressed in the development of different lymphomas, including follicular lymphoma, due to a translocation between chromosomes 14 and 18. Genes that promote apoptosis, we've talked about one of these already, which is p53. Another which of these is BAX. We now move on to how these cells that have dysregulation both in the development of their oncogenes, their tumor suppressor genes, and alterations in genes that regulate apoptosis undergo development of clonal expansion. What we have in this cartoon is simply a diagram that tries to help you understand the development of clonality. You have the development of a female zygote from the sex chromosomes of the mother and the father. Note that in the blastocyst there is inactivation of one X chromosome. Thus, in the development of neoplasia in the adult female, the neoplasm can either be polyclonal, where in some cells the mother's X chromosome has been activated, and in others the father's X chromosome has been inactivated. In the majority of cells in the body, in order to maintain a balance in terms of the amount of signaling coming from the two X chromosomes, predominantly the father's X chromosome has been inactivated, leaving only the mother's active. This is this middle population of predominantly red cells. If just the father's X chromosome is present and the mother's X chromosome has been inactivated, this leads to another potentially monoclonal neoplasm. Finally, how do tumors progress? The best example of this is seen in the GI system, where we can carefully diagram the progression from an adenoma to a carcinoma. Here what we're trying to demonstrate in this cartoon are the mucosal changes that lead to the development of carcinoma with the underlying genetic changes listed below. First of all, remember Knudson's two-hit hypothesis, in that we have to have various changes within the germline that can be either inherited and or somatic, that knocks out our various tumor suppressor genes. This is our first hit. An example of the sort of gene that would be knocked out is the APC gene on the long arm of chromosome 5. You now have an area of mucosa that can be considered at risk. There now is the need for a second hit. Sometimes this is inactivation of normal alleles through things such as methylation abnormalities. An example here would be changes in genes such as beta-catenin. You now have proto-oncogene mutations such as KRAS on the short arm of chromosome 12 that lead to the development of the premalignant lesion such as the tubular villus or tubulovillous adenoma. You now have the homozygous loss of additional cancer tumor suppressor genes, overexpression of genes such as COX-2, loss of tumor suppressor genes such as p53. This leads to the beginning of an actual formal carcinoma, as you see represented in blue in the last part of this slide, which now can invade through the muscularis and metastasize to other parts of the body. Now, the body always has a constant outlook for cells that have developed a cancer phenotype, and thus one of the things that must also occur is a loss of immune function. Immunosurveillance normally destroys neoplastic cells via recognition of non-self antigens. Humoral and cell-mediated immune responses both play a role. Thus, patients with immune system dysfunction are going to have an increased number of neoplasms. In terms of metastases, what needs to happen is that the neoplastic cells not only move their way past any sort of basement membrane or other limiting membranes in the surrounding tissue, but they need to make their way either to the blood vessels or to the lymphoid vessels to find a way to seed elsewhere in the body. In terms of diagnosing cancer, there are various immunohistochemical stains that we can use to try and diagnose what is the cell of origin of the particular tumor that we're looking at. Epithelial cells tend to express cytokeratins, hence we can use immunohistochemical stains for different high and low molecular weight cytokeratins to determine if the cell of origin is in epithelial cells. Muscle cells can be delineated by the use of markers such as desmin or smooth muscle actin. Mesenchymal cells can show expression of the marker vimentin. Some organ systems have markers that are specific to them, such as prostate-specific antigen, or the clusters of differentiation, or CD markers, that are associated with the development of lymphoma and leukemias. Breast cancers are infamous for expressing estrogen and progesterone receptors. S100 is seen in the expression of tumors such as melanoma and neural tumors, and thyroglobulin is a particular marker for thyroid cancer. That ends this lecture in the Falcon Pathology Review Series for USMLE Step 1 on neoplasia.